Hello, students. I welcome you to the last lecture of the lecture series on cell culture technology. This is Dr. Joyjyoti Das, Assistant Professor of Biotechnology at Amos University. So far, we had discussed about the essence of cell culture, what uh, cell culture essentially means, and what is its various applications in the medicine and health sector. We have learned about what are the design considerations for an animal cell culture laboratory, what are the various commonly used equipment and reagents in an animal cell culture laboratory, and we have also discussed in detail about the media uh, used in animal cell culture. And we have also uh, talked about the usual contamination that occurs in animal cell culture. So, in this final lecture of the series, we are going to further talk on certain important practices of routine animal cell culture and a knowledge on these few topics are going to equip you with the basic uh, knowledge and know-how of animal cell culture practices. So, first, we'll talk about the principle and practices of the technique called cryopreservation. Then we will talk about how to quantify cells or how to estimate the number of cells in a cell suspension. And finally, we are going to talk about an important analysis with cells, which is called as cytotoxicity analysis. So let's start with topic cryopreservation. So, what is cryopreservation? So, as I've talked or as I've mentioned to you earlier, that there are cell repositories uh, uh, in this world where you can actually borrow cells from to work in your laboratory, borrow or purchase cells from to work with in your laboratory. So, how are those repositories, cell repositories, maintaining the cells? Are they growing all the cells together and continuously subculturing them? So there are so many different uh, types of cells. Uh, is it humanly possible to culture all the uh, cells together simultaneously? Or is there any method by which you can preserve the cells and again revive them in culture as and when required? So, that particular technique is a very important technique when it comes to animal cell culture, and it is called as cryopreservation. So, cryopreservation is a non lethal storage of biological material at ultra low temperature. So, what temperature range we're talking of? We're talking of a temperature of minus 196 degrees Celsius, where almost all metabolic activities of cells cease. And in such a state, the sample can be preserved for extended periods of time. However, only few biological materials can be frozen without affecting the cell viability. So we are going to talk about that. So why would you freeze cells? Of course, as I mentioned, for long-term storage. That is the first or one of the main applications or rationale behind freezing of cells. But then, if you maintain a cells in culture over a long period of time, there might be issues of genotypic drift due to genetic instability. And as I had mentioned in one of the previous lectures, that genetic instability is a naturally occurring phenomena due to the due to random mutations, and there might be changes in genotypic characteristics or phenotypic characteristics of cells if we're maintaining them in culture for a long time. And senescence and the resultant extinction of the cell line. So if you're working with primary cells, we have discussed this in detail previously, that if you're working with primary cells of a normal tissue, they will have a finite lifespan. And once you have isolated them from living tissue, you would like to complete your entire set of experiments using those cells. And so it is prudent to store sufficient amount of that cells uh, before you start the experiment, so that you can end all your runs of the experiment with the same stock of cells for homogeneity of results. Then there might be transformation of growth characteristics and acquisition of malignancy-associated properties. So this is again a transformation that might occur in your cells due to random mutations. Similarly, for phenotypic instability, and the most common factor uh, that can occur across all labs that are routinely culturing cells is contamination by microorganisms. So you procured a cell line or a group of primary cells, maybe from some laboratory, and you have started working with it, but you forgot to preserve a copy of the same, and suddenly your cells get contaminated. Then your effort goes in vain, and you need to repeat it all over again. Uh, Plus, we know that not only microbial contamination, there's a chance of cross-contamination by other cell lines if you're handling too many together. So misidentification due to careless handling, that can also lead to mixing of cell lines. Incubator failure, that might happen when the CO2 runs out, or there are any other technical issues with the incubator. Saving time and materials by not maintaining lines other than those in current use. Of course, why will you do so? Because uh, reagents such as media are costly, and you wouldn't want to spend too much on routine maintenance of cell cultures, right? So, and also the uh, need for distribution to other users, because since the cell repositories are maintaining the cells and routinely cryopreserving them, so when the researchers are applying to them for a cell line, they can send them a copy of it. So, and how do we perform cryopreservation? What reagent is used, or what uh, chemical is used? So, liquid nitrogen is the most widely used material for cryopreservation. Dry ice can also be used, but liquid nitrogen is more effective and routinely used in cell culture labs. So so how is liquid nitrogen stored in the lab? It is stored in the form of stored in containers called as cryo cans. Okay? And what you can see in this image here is an opened cryo can, from which you can see fumes of liquid nitrogen emanating out, and this pair of tweezers is holding a cryo vial, which contains your cells in certain media that we are going to talk about in the next few slides. So why would you use liquid nitrogen? Because it is chemically inert, it is of relatively low cost, it is non-toxic, non-flammable, and readily available, because uh, the majority of air contains nitrogen. We all know that from school level. So how to go about cell freezing? What is the theoretical background? Optimal freezing of cells for maximum viable recovery on time depends on minimizing intracellular ice crystal formation and reducing cryogenic damage from high concentration solutes found when intercellular water freezes. So what does this mean? What does this essentially mean? It means that to freeze your cells, to take your cells to such low temperature, you need to keep one very important thing in mind, and that is while uh, transformation of the state from a state of water or media from liquid to solid, there might be issues such as ice crystal formation. And since your cells or biological material contains water, there is a high chance that the water, while transforming its state, may form ice crystals. And if there is formation of ice crystals within the cells, of course, and cells are soft materials and they contain organelles and proteins, etc. So the ice crystals might uh, rupture not only the membranes of the cell itself, but also the membranes of the intracellular organelles, and that would destroy the cell. So how to achieve cell freezing in an optimal manner? So it is achieved by freezing slowly to allow water to leave the cell, but not so slowly that ice crystal growth is encouraged. And to aid this process, you require a hydrophilic cryoprotectant to sequester water. So that cryoprotectant is hydrophilic in nature and it can draw water out of the cell while itself occupying the it, occupying the cell, entering the cell. So, and then once you have done this process, you store the cells at the lowest possible temperature. And for that, you are using liquid nitrogen, while again reviving the cells from that frozen state. And that process will be called as thawing. So while thawing, you have to do this process of thawing very rapidly to again minimize ice crystal growth, as the frozen material inside the cryovial will start warming up, and thereby rapid thawing will allow or avoid rather the generation of solute gradients as the residual intracellular ice would melt. So what are cryoprotectants? Uh, Cryoprotectants, by definition, these simply increase the total concentration of all solutes in the system. They reduce the amount of ice formed at any given temperature, but to be biologically acceptable, they must be able to penetrate into the cells and exert least possible toxicity to the cells. Okay, so we are going to discuss more on that. So what is the role exactly of cryoprotectant? Why are you adding cryoprotectant while taking your cells to a very low temperature? There are two potential sources of cell damage during cryopreservation. One, as I already discussed, the formation of large ice crystals inside the cell that may rupture membranes, and intracellular concentration of solutes increase to toxic levels before or during freezing as a result of dehydration. Okay, so cryoprotectants here will act like an antifreeze, and they will lower the freezing temperature while increasing the viscosity. While increasing the viscosity of the environment inside of the entire liquid inside the cells. Okay, and they will prevent any cellular damage in the process. Okay. So, uh, so how to constitute a freezing medium? Will you add directly? Will you add DMSO or any other cryoprotectant directly to the cell for cryopreservation? No. So you have to constitute a medium first, which is called the freezing medium, and it comprises a standard formulation for a freezing medium. 10 ml is that it comprises 5 ml of fetal bovine serum, 4 ml of the basal medium in which the cell is cultured, and 1 ml of DMSO. So that's around 10% DMSO. So the cell suspension is frozen in the presence of a cryoprotectant such as glycerol or dimethyl sulfoxide. As in case of microbial cells, you would use glycerol for cryopreservation. Here, in animal cell culture, the most commonly used cryoprotectant is dimethyl sulfoxide. And of these two mentioned here, glycerol and DMSO, DMSO appears to be the more effective, possibly because it penetrates the cell better than glycerol. Okay. So concentrations of between 5 and 15% have been used, but 7.5 or 10% is more usual, which is why I've given this as a standard formulation for freezing media. Okay, so how should the process of freezing be? We already discussed that the process of thawing a cryopreserved stock of cells should be rapid, but for freezing, it is exactly opposite. So the preferred method of freezing cells using the freezing media is the slow freezing method. So it has been seen the tissue is slowly frozen. If the tissue is slowly frozen with decreasing temperature of say minus 0.5 to 5 degrees Celsius per minute from 0 to minus 100 and then transferred to liquid nitrogen, then the tissue or the biological sample of interest will stay put or it will stay intact for extended period of time. So most cultured cells survive best if they're cooled at 1 degree Celsius per minute. So, which is why after adding the freezing media to your cells of interest, you should put the stock in uh, 0 degrees Celsius, and then after maybe uh, 15 minutes or half an hour, you could take it to minus 20 degrees Celsius freezer and keep the cells at minus 20 degrees Celsius freezer overnight. And maybe the next day you can transfer the stock to minus 80 degrees Celsius. And maybe 24 hours after that, you should transfer it to your cryocan, which will have a temperature of minus 196 degrees Celsius. So this slow freezing method will minimize ice crystal formation as the entire cryoprotectant and gets frozen and the cells can be stored in minus 196 for long periods of time. And a minimum cytotoxicity will be observed. So the goal in cryopreservation is to prevent formation of ice crystals as you take your cells to the ultra low temperature of minus 196 degrees Celsius, so as to minimize cell loss due to the freezing procedure. So storage, we have already discussed it. I have already discussed that the transition should be gradual from 0 degree to minus 100 degree. The transition should be gradual and then should transfer it to minus 196 degrees Celsius. And again, for thawing, rapid thawing is required because rapid heating will prevent localized recrystallization during thawing as the temperature increases to 0 degrees Celsius from minus 196. So if the procedure is done quickly, then there is a less chance for cells to lose their viability due to ice crystal formation. Okay, so what can be cryopreserved? In general, cryopreservation is easier for thin samples and small clumps of individual cells because these can be cooled more quickly and so require lower doses of toxic cryoprotectants. Therefore, the goal. So, in general, DMSO is toxic to cells, but it is of it exerts low toxicity to cells, and which is why it has been used as a cryoprotectant. The goal of cryopreserving human livers and hearts for storage and transplant is still some distance away because they are thicker tissues and there are issues uh, while freezing these kind of thick tissues. The cryoprotectant cannot enter properly into the tissue, and in the process, the uh, entire process fails. Nevertheless, suitable combinations of cryoprotectants and regimes of cooling and rinsing during warming often allow the successful cryopreservation of biological materials, particularly cell suspensions or thin tissue samples like semen, blood, stem cell, umbilical cord blood, egg, embryo, plant, etc. So the applications will, of course, include freezing of cell cultures, maintenance of disease-free stock of cells or organisms, seed bank, gene bank, and storage of rare germ plasm. And we have also seen applications of cryopreservation in various sci-fi movies, and it is already known that humans are making research advances in this field to store even humans and revive them back in future. So while the benefits are the use are that it is cryopreservation is useful in the breeding of dairy, cattle, pigs, and dogs, and storage of cancer and tumor cells, storage of stem cells, umbilical cord blood cells, uh, even sperm and ovum cells for the process of in vitro fertilization. But then the disadvantages are that there is a slight involvement of cost in it because you need to run a cryopreservation unit, that is a liquid nitrogen production unit, so that you can procure liquid nitrogen time to time. And there are social issues of preserving uh, reproductive cells such as sperm and ovum also, which is one of the main issues with the process of in vitro fertilization. However, it's a very important technique when it goes to animal cell culture, and which is why it is an important part of today's lecture. So now we come to the next important practice in cell culture, which is manual quantitation of cells. So uh, how do you quantify your cells? How you have trypsinized cells from a flask and you have centrifuged the cells, you have a cell pellet, but what is the number of cells in that cell pellet? Of course, it is not possible to count one by one unless you have a systematic uh, device that can enable counting of a lesser number of cells, and then you can perhaps estimate the original number of cells in the suspension by unitary method. So that is the principle on which your manual device for cell counting works. And what we usually use in cell culture labs is called a Neubauer hemocytometer. So what does it have? So I'm showing here with the arrow this particular portion of the device. So this is a glass-based device, and these two portions of the device are which contains the uh, counting grids, cell counting grids, which I'll show you in the next slide. So the grids look like this. So what are the characteristics of these grids and how are they special? The hemocytometer is divided into 9 such major squares of 1 millimeter by 1 millimeter size. The 4 corner squares identified by the red square here. So I'm marking it with an arrow for your ease of understanding. 4 corner squares are further subdivided into 4 grids. So The smallest square is this one, and 4 by 4, that is 16 such small squares, one will make one corner squares. And there are 4 such corner squares. The height, so basically, if I go back to the previous slide, so this on this device, you put a cover slip and you make this a chamber. So this is a grid that we saw here. And if you're placing a cover slip here, then it will not only have a length and breadth, but it will also have a slight height. Okay, and that height is 0.1 millimeter, while the width and length are 1 millimeter each. So what will be the volume of the chamber formed if the cover slip is placed on top of the grid? The volume will be 1 millimeter by 1 millimeter by 0.1 millimeter, and that will be equal to minus 10 to the power minus 4 ml. So the volume of one corner square is 10 to the power minus 4 ml. So that is what we need to keep in mind for our calculation. So what do you need to do next? How do you need to use the device? So you have to take your cell suspension uh, using a pipette, and say a 20 microliter pipette or a 200 microliter pipette. And since the cover slip is lying here on top of this grid chamber, so you have to push in or infuse the cell suspension through your micropipette into this particular chamber. So what will happen? Cells will be spread all over this chamber. All over this chamber, cells will be spread. And now you have to count the number of cells that are spread over this particular chamber, because you know the dimension or the volume of this particular chamber. So if you count the number of cells that have spread over this particular chamber, then by unitary method, you can find out how many cells must have been present per ml of your cell suspension. So that is the simple logic to that goes into like counting cells using a hemocytometer. Okay, so what will be your final formula? Your final formula will be total number of cells per ml of your suspension will be the average cell count per square into dilution factor into 10 to the power 4. So before we come to that, I need to explain this part, why there are 4 squares here. So 4 corner squares are identical, and so how would you get your reading? So you have to count all the 4 corner squares shown in red here. Okay. And while counting, uh, you need to keep a thing in mind that the cells which are in the center of each small square should be taken into consideration. But what about cells that are lying in the edges of any of these squares? So in that case, you take any 2 adjacent sides of the small square. Okay, so if you're taking top and left, take top and left of every square. So in that way, you will count one circle only once. So these circles are cells. So if you take convention that you will count the cells which are touching the top or left of any square, then you will be avoiding duplicate counting of cells. So in that way, you count all the 4 corner squares and then you average the value. Okay, so that's for like that's for averaging your value and getting perfect reading for cell counting. Okay, so that's why the formula says total number of cells per ml will be average cell count per square into 10 to the power 4. 10 to the power 4, because the volume of one square was 10 to the power minus 4 ml. So from there, the formula came as average cell count per square into 10 to the power 4. Why is this dilution factor coming here? Because suppose you have taken a cell suspension and you have loaded cells directly into the hemocytometer, then this formula will essentially translate to total number of cells per ml of your suspension will be average cell count per square into 10 to the power 4 directly. But in case your cell suspension was already diluted from an original stock, then how many times you diluted and then transferred it to a hemocytometer, that number of times will come as your dilution factor. And that needs to be multiplied to get the cell suspension in the original uh, stock of cells. Okay, hope that is understood. So how can you use this for cytotoxicity analysis? Say you have added a drug to your cells, to your group of cancer cells. You're testing your drug on a group of cancer cells, and you want to compare it with your control sample. And your control will contain only cancer cells. Your experiment will contain cancer cells treated with the drug. You want to compare the uh, level of cytotoxicity of the drug. How will you do it? You will take cells from both the wells, and then you will perform this specific assay. What is this assay? You will use the hemocytometer for counting cells from both the wells. But before that, you need to stain the cells with this particular dye, which is talked about in this slide. So you use a trypan blue dye, which is a vital dye, and this and works on the principle of dye exclusion. So if your cells are live, their membranes will be intact, and this particular dye, trypan blue, will not be able to enter into the uh, live cells. Okay, but if your cells are dead, then the membrane will be leaky, and your cells and these trypan blue dye will be able to enter into the cells, and so those cells will appear bluish in color. So now if you load the cells on the hemocytometer, you will see something like this. Okay, there will be white cells, which are your live cells, and there will be some dead cells, which will indicate your uh, there will be some blue cells which will indicate the dead cells in your sample. So remember, the white cells are your live cells and the blue cells are your dead cells. So uh, just for the sake of understanding, you can pause the video for a while and you can calculate the number of live cells and dead cells per ml of the cell suspension by looking at this diagram. Okay, so now let's move to one last technique, which is also routinely performed in a cell culture lab. So just now I talked about the manual counting procedure of cells, and that you can do inside your cell culture room itself, where you have kept the microscope for routine observation. So in that particular microscope only, you will be using the hemocytometer to count your cells and passage them accordingly, or use certain requisite number of cells for a certain experiment of your cell culture. But there is also another method wherein you won't be performing a manual quantitation of cell. Instead, you would do quantitation of cells using the help of an instrument, and that instrument will be a UV-based spectrophotometer. So how can you perform that experiment? So for that, you need a special dye called an MTT, and which is why this assay is called as an MTT assay. MTT assay is used to measure cellular metabolic activity as an indicator of cell viability, proliferation, and cytotoxicity. This colorimetric assay is based on the reduction of a yellow dye, which is the MTT itself, tetrazolium salt 3,4,5-dimethyl thiazole 2,5-diphenyl tetrazolium bromide, or MTT in short. So This dye, when it enters the living cell, the living cell can reduce this dye by help of its uh, different oxidoreductase enzyme to form purple formazine crystals. Okay. And this is only possible if the cell is metabolically active, which means if the cell is live. If MGT enters a dead cell, it won't be converted to the purple formazine form, right? So once you have incubated your cells with MTT, you then remove all the cell and the media from the wells, and because the formazine crystals will be sticking to the surface of the wells. So generally, we perform these type of assays in 96 well plates, and we generate triplicate data, and we generate data for different samples of varying concentrations on the same plate for comparative analysis. So once the formazine crystals are formed, we solubilize the formazine, adding DMSO as a solvent for formazine. And then a purple colored solution will result, which you need to take the absorbance reading of at 595 nanometers using the UV spec UV-based spectrophotometer. So this plate here shows a sample of such an MTT assay, and in this plate only we can see uh, an increasing gradient of formazine crystal. I mean, increasing gradient of purple coloration, which means increasing gradient of formazine crystal formation, which means that there were less or no viable cells in these wells, and viability of the cells kept on increasing as you go from left to right. Hope you have been able to understand the principles and practices of cryopreservation, cell counting, manual cell counting, and cytotoxicity analysis. Thanks for watching the video. Hope the lecture series have enlightened you about the basics and principles and practices of animal cell culture technology. Thank you.